In this section, we're going to start by talking about what a genome is, and then we're going to move into how a genotype creates a phenotype. So a genome is all of the genes, all of an individual's genes. So the human genome, when we talk about that, what we're saying is all of the genes present in a human being. The human genome contains about 20,500 genes, made up of about 3 billion base pairs. So that's a lot of DNA. And if you took all of the DNA out of a single cell and laid it end to end, it could stretch all the way to the moon or something like that. When we talk about genomes, we actually measure them not in billions of base pairs, but in millions of base pairs. So in this diagram, we show the human genome, about 3.2 billion or 3,200 million base pairs. That's what the MB is for. And you can see that dogs have a smaller genome. They have 2,400 million base pairs. Chickens have an even smaller genome with 1,200 million base pairs. And this may lead you to think, humans are more complex. Therefore, we have a bigger genome. And that is what the first researchers, when people started figuring out like how big genomes are, they're like, humans, we must have a really, really big genome. And then they did some more research and they found that a white spruce tree has a genome 23.6 billion with a B, base pairs. A red spotted newt, which is an amphibian, 39.1 billion base pairs, and a lungfish, which lungfishes are mostly haven't changed a lot since some of the first fish that walked on land. Lungfish have, as it sounds, they have gills and lungs, and they can breathe air. They have a genome that is 130 billion base pairs, or 130,000 million base pairs. So it does not hold that things that seem to us to be simpler have smaller genomes. Genomes are complicated. And one of the ways that works is goes back to the thing I said in my last section, which was that not every gene codes for just one protein. And that's one of the ways that our genome is able to be smaller than you might expect. All right, so how does this collection of genes become the phenotype that is, for example, an individual human or an individual tree? It's a two-step process in which a gene is first transcribed from DNA into RNA and then translated from RNA into a protein. And if you're thinking, hang on though, but organisms are more than just proteins. They're also like fats and things like that. Yes, but all of those things that make up organisms get made by proteins. So you got to have proteins. Wild, right? Okay. This can be oversimplified, this DNA to RNA to protein, as something we call the central dogma. I do not like teaching the central dogma because it is in and of itself actually incorrect, but it's a thing that people talk about. So you got to at least know that it's a thing. The central dogma says that DNA is transcribed into RNA and RNA is translated into proteins. Okay, DNA, RNA, protein, to shorten it to its shortest. This is actually a misunderstanding of what was first meant by the person who came up with this phrase central dogma, which is amusing because a dogma is actually a set of beliefs that are not actually correct. So the fact that the central dogma is actually not understood correctly is weirdly apropos. So what am I talking about? Well, the person who came up with this was Francis Crick, who was one of the people credited with discovering the structure of DNA. And he was thinking about this process of information flow from DNA to RNA to proteins. And he said, what the central dogma is that information can go from DNA to RNA, and sometimes it can go from RNA back to DNA. Like you could take a strand of RNA, you could copy the exact strand of DNA that created that strand of RNA. But once you get to proteins, you cannot copy from a protein back to the original RNA or DNA that created that protein. And in a minute, you'll see why that is. So the information can go from DNA to RNA, it can go from RNA to proteins. It can go from RNA back to DNA, but it cannot go from protein back to RNA or DNA. That's what he meant. So why did he call it the central dogma? He actually didn't know what the word dogma meant. He just thought it sounded really cool. And he was giving a presentation. So he's like, here's my central dogma. And everybody was like, cool. Because they didn't stop and say, maybe Francis Crick is using the wrong flippin' word, which he was. So there. This annoys me because science is hard enough when we use the correct words, when people are throwing around the wrong words, like, oh my God, why are you trying to kill us? Okay, so how do we do this? DNA to RNA to proteins. Okay, so we start off, the DNA strand is transcribed into RNA, and that just means that a strand of RNA that is complementary to the DNA is going to get made. Okay, so transcription, think of it as if you transcribe a video, you listen to the video and you make a written record of the words in the video. So it's the same language, just in a slightly different form. So we're going from DNA, which has the deoxyribose sugar, to RNA, which has the ribose sugar. Same language, still nucleotides, just a slightly different form. So RNA polymerase is the molecule that's going to come along. It's going to bind to DNA, open up the double helix, and then start inserting RNA nucleotides to complementary bind to the DNA nucleotides. Now remember, RNA doesn't have the same nucleotides as DNA. It does have cysteine, guanine, adenine, but instead of thymine, it has uracil. So we take the strand of DNA. If there's a T, the RNA that gets attached is an A. If there's an A in the DNA, what gets attached to it is a uracil. Okay, because normally if it were DNA, it would be A to T, right? In this case, it's A to U. From G to C, G binds with C, G binds with C, C binds with G. The rest of it's normal. It's just this A to U part that you need to remember, okay? So RNA polymerase comes along, binds at a promoter, which is a specific DNA sequence at the beginning of a gene. And then there's this 3-step process, which is just initiation. The RNA polymerase attaches to the promoter and starts transcription. Elongation as it continues, and then termination, the RNA polymerase gets to another DNA sequence called a terminator that tells RNA polymerase that was the end of the gene, you can let go now. So that looks like this. Here's our gene, okay, with the promoter at one end and the terminator at the other. RNA polymerase comes along, binds to the promoter, opens up the DNA, and starts reading the DNA and creating a complementary base paired strand of RNA. It reads all the way along until it gets to the terminator, and then it lets go, and it lets go of the RNA as well. So this RNA is going to have the complementary base pairs to this strand of DNA. And if you're thinking, hang on, but it's only one of the parts of it, not both parts of the double helix, you are correct. So it reads one strand of the double helix, not both of them. All right, now, when that is finished, this RNA strand is not done. It has to go through a couple more steps in order to be made into something that can go out of the nucleus into the cytoplasm to be translated. Okay, remember, the chromosomes are inside the nucleus, so transcription happens in the nucleus. So the first thing that happens is that a cap and tail are added. And these are both just segments of RNA that signal to the nuclear envelope that it's okay to let this strand of RNA out of the nucleus. and also signal to the ribosomes in the cytoplasm, hey, this is something you bind to. And then the tail are like just extra RNAs. It's called a poly-A tail. It's just a bunch of adenines at the end, just to signal to things and to tell the ribosome you're done. Okay. Introns have to be removed. And this is a little bit of a head scratcher. So it turns out that, if we go back here, our gene here, not all of it gets used to make a protein. There are a bunch of sections that are like extra. They're called non-coding sections because they're not part of the instructions for making this specific protein. We got to take those out. The coding regions that are going to stay in this strand of RNA, they're called exons. And to me, these are backwards because the exon should be the one that's getting removed, but it's not. And so the way I remember it is the exons are the ones that exit the nucleus. Okay. The introns are the non-coding portions, so they stay in the nucleus. That's the only way I can remember which is which. All right. All of this, the adding the tail, the removal of the introns and the connecting of the exons is called RNA splicing. And splicing is just where you take a segment out of something and you put the remaining ends together. When we're done with all of that, then we have what we call messenger RNA, or mRNA, which is ready to go out into the cytoplasm. Okay, so this is what that looks like. So here's our strand of DNA in blue. There's a promoter here, a terminator here. We've got exons and introns. The RNA polymerase transcribes the whole thing. It doesn't care what's supposed to get used or not. All it does is just complementary base pair. Okay, it doesn't do anything else. Like, I'm just copying. Don't ask me what's supposed to be here. So here's our strand of RNA. The cap and tail have been put on. And then these intron sections, they get cut out. There's another protein that does that. And the ends get spliced back together. And now we have our strand of mRNA. And that then leaves the nucleus and goes out into the cytoplasm. Okay? So DNA to RNA transcription, that's that process. Then we have RNA processing, including RNA splicing. Now we're ready to make a protein. Okay, I'm going to come back in just a second and learn about how we do that.